ABC model is a model of flower development based on the expression of three homeotic gene groups, group A, B, and C. Flowers consist of four whorls of floral organs that develop in rings that flank the meristem. The first to develop is the sepal whorl, followed by the petal whorl, then the stamen whorl, ending with the carpel whorl that stays the central whorl. This model of flower development has been studied in the model plant species Arabidopsis thaliana. In Arabidopsis, the A genes are represented by two main genes, Apitella 1 and Apitella 2. The B genes are also represented by two main genes, Apitella 3 and Pistillata. The C genes are represented by one gene, Agamus. These genes work together in the development of the different flower whorls. A gene expression alone is responsible for the development of the sepal whorl. Both A gene and B gene expression together are responsible for the development of the petal whorl. Both B gene and C gene expression together are responsible for the development of the stamen whorl. C gene expression alone is responsible for the development of the carpel whorl. Mutations in any of these genes affects the development of these whorls. A wild-type flower with no mutations in any of the gene groups will present with all four flower whorls. Mutations in the A genes affects the two outermost whorls, the sepals and the petals. In mutants lacking A genes, no sepals or petals develop, as both these require the expression of A genes either alone or in conjunction with B genes to develop. Instead, C genes are expressed instead of the A genes. This means that in place of the sepals, carpels instead develop as the first whorl. It also means that in place of petals, stamens develop as the second whorl. The third whorl stays unchanged and still develops as stamens, as does the fourth whorl that still develops as the carpels. In plants with mutations in the B genes, no petals or stamens develop, as both these require the expression of B genes in conjunction with other genes to develop. As in wild-type plants, the first whorl to develop has not been affected by this mutation and still develops as the sepals. But because no B genes are expressed, a second whorl of sepals develops as the second whorl in place of what would have been the petal whorl. Instead of stamens, the third whorl develops into a carpel whorl. The fourth whorl stays unchanged and still develops as the carpels. In plants with mutations in the C genes, no reproductive organs develop, so no stamens or carpels develop as they require the expression of C genes to develop. A genes are expressed instead of the C genes. In these plants, whorl 1 is unaffected by the mutation and still develops as sepals. The second whorl is also unaffected by the mutation and still develops as petals. But because A genes are expressed instead of the C genes in this mutation, the third whorl develops as a second whorl of petals instead of the stamens. Whorl 4 is also affected by this mutation, and instead of developing into the carpels, it develops into a second whorl of sepals. Thank you for